Okay, so I think we're going to start. First of all, thank you for joining us for this afternoon session. And I don't know how many of you were in the morning session. I have to remember to stay close to the microphone. How many of you were in the morning session, in the workshop? Did you enjoy it? Hopefully, we're going to be as entertaining and informative as the morning session. So, my name is Piero. I am the president of the society. So on behalf of the society, I first would like to welcome all of you to the 2024 Congress for In Vitro Biology. We are excited to be here in St. Louis. We are excited to see so many of you attending this conference this year. And what we decided to do with this workshop, first of all, this is a two-part workshop. So we're going to have the first part this year in 2024, and then in 2025, we're going to have the second part. This workshop is supported by a grant by the National Science Foundation. And the idea behind the workshop is really what is captured in the title. We really want to go back to the basic. We realize as a society that there is a knowledge gap when it comes to the basic understanding of media formulation. And I always think of it as when you start to learn your alphabet. You learn your letters, then you start to compose words, and then you start to be able to write full sentences. This is exactly what we would like to accomplish today, to share with you that alphabet, those letters that we think have been within time somehow lost just because right now there is a lot of words and sentences in the media that they are already written on your behalf, and we don't really are able to correct when and if needed and troubleshoot because we don't have a complete understanding of what are the basic components of the media. So as you see in here, I'm going to give a quick introduction, and then we're going to have three experts in the field that they're going to share with us insight of media formulation. The first person is going to be Chuck Armstrong. He is currently a plastomic, and he's going to give us an overview, a general overview of media components. Then we're going to have Ken Torres. And if you are in plant tissue culture, I'm sure that the name Phytotech Labs means something to you. Well, Ken is the person behind Phytotech Labs. And he's going to give us an overview of media preparation and quality. And then we're going to have with Max Jones from the University of Guelph a look in what we consider is going to be the future of media formulation with use of design of experiment, AI for media optimization. I just want to remind all of you that this session is going to be also recorded, and then it's going to be made available on the Society YouTube channel. So feel free to take pictures today, but don't forget that the video recording of this session is going to be available on the Society channel. One thing that we just noticed as we were on stage, and this speaks also for what is the Society. We have on stage members from different years of membership. I have been with the society for 15 years, Max 5, Chuck 25, and Ken 35. So this is exactly one of the strengths of this society, where you have generation of scientists that they are sharing knowledge and feeding their knowledge into each other. And this is exactly what we're going to do today. So now, without any further ado, I'm going to give you a quick introduction, and I am quoting Trevor Thorpe. If you don't know Trevor Thorpe, he was a very active member of the society, and actually starting this year, we're going to have every year a session that is going to be held in his memory. Trevor was a very active member of the society, was a pioneer in the plant tissue culture field. And what you see in this quote is a definition of plant tissue culture. Plant tissue culture is the aseptic culture of cells, tissues, and organs and their components under defined physical and chemical condition in vitro. And this is exactly what we're going to address today, the chemical condition that enable the plant tissue culture. Now, I'm going to give you a quick timeline just to put in perspective how media formulation and tissue culture develop within time. I'm not going to spend that much time on this slide, but this is just to remind everybody that we really start to think of the cell as the basic unit of life around 1838. And that's when you have the first really mention of the concept of cell. And every plant and animal shouldn't, this is a quote, is an aggregate of individualized, independent, separate things. As you see then, within time, the cell theory evolved, and we are now really in 1858 describing tissue as the sum of individual cells. Now, when it comes to plant tissue culture, really in 1908 is the first time that we really have what can be considered a definition of plant tissue culture. What you see here is the work from Heberland that was able, using leaf mesophyll cell from a red nettle, that were placed in culture, in hydroponic culture, and they were he was able to sustain their growth for up to 1 month. So this is the real first report of what can be considered plant tissue culture. And then, of course, from there we go to the experiment of Philip White in 1934 that used tomato root tips. And what White was able to do, he was able to subculture up to 52 times those root tips, demonstrated that indeed the growth that he was observing was due to the supply of nutrients that he was providing using the media. From there, of course, then you have additional experiments that validate the ability of tissues, plant tissue, to grow if provided the right nutrients and the right condition. And then in there, you see in this slide, I also started to put as a reference for you when indolic 3-acetic acid was identified in 1936. And then in 1955, you had the discovery of kinetin. So the reason why I brought them there is because that helped us to put in perspective from the time point of view when plant regulator really started to be discovered, understand, and then used for tissue culture application. And then, of course, you start to have in 57 the work of Skog and Miller that were able to, playing with the concentration of indolic acid and kinetin, they were able to differentiate root and shoot. And then from there, you start to have the work that then ended up to be in the development of the MS media, the Murashige School in 1962. Media that is one of the basal media that is very commonly used in tissue culture. From there, you can start to see the development of different media formulation, basal media formulation from the LSM medium in 65, the Gamborg B5 media in 68, and so on. So this is, again, just to give you a very general overview of the development of the media formulation and how media formulation went hand-to-hand with the specific needs of specific crops and specific application. So this is my introduction. Now I'm going to pass the microphone to Chuck. We envision this workshop to be interactive. So there are slides. Chuck, Ken, and Max are going to present. But if you have questions, we're going to open the floor to the questions, and then we're going to address them as we proceed. Okay? So thank you.
Okay, good afternoon and welcome to St. Louis. I've been living in St. Louis for over 35 years now, 33 years at Monsanto Bayer, now 3 years at Plastomics. So it's a nice city. Those of you out of town, hope you enjoy it here. Happy to be a part of the session this afternoon. So I put this up here. How many of you have ever mouth pipetted stock solutions? Anybody? A few? Okay, a few. So, yeah, no safety people here, right? So back when I was a grad student at University of Minnesota in the early 1980s, I mean, that was common practice, mouth pipetting, stock solutions. And Dave Duncan, a good friend of mine, used to joke that mouth pipetting, you're getting a mouthful of 2,4-D is what made us who we are today, for better or worse. We don't do that anymore. I assume nobody's doing that anymore. But one thing we were doing back at that time too, though, I think was a better thing, perhaps. We were making our media from scratch, making our stock solutions from scratch, and we pretty much had to know what was in it then. So now, I doubt very many of us do that anymore. We've got the prepared media formulations that we just buy from Phytotech or Sigma or Amazon even, and it helps a lot. I mean, it's a great tool to have, but as part of that, we kind of tend to forget what's in those. So part of this session now is just a reminder what's in these bottles of media that we're buying and using all the time. So might be a little too basic, we'll see. I'm going to try to add in a few things here and there, just on a few components, the tidbits of interest that might be useful. I want to give Ken a lot of acknowledgement for the slides I'm going to use. He put a great deck together that Phytotech used. There's like 180 slides, I think. So it's been boiled down quite a bit, and hopefully there'll be something useful here. Okay, so they can be broken down in various ways. This is the way we'll be going through these today. We've got our macronutrients, micronutrients, vitamins, and so forth. Start with the macronutrients. Okay, so basically 6 major elements without which plants can't grow. They've been defined by the International Association for Plant Physiology as things that are present as greater than 0.5 millimolar. Again, you can categorize these things different ways, but this is how we're doing it for this particular purpose. So those 6 are nitrogen, potassium, phosphorus, calcium, magnesium, and sulfur. Especially nitrogen and potassium, those are the ones that are in the greatest concentrations, over 20 millimolar. The other 4 down in the 1 to 3 millimolar range, found in all the plant media that we used, also found in a lot of commercial fertilizers. We'll go through those briefly first. Nitrogen, probably one of the most important macronutrients that's included in our media. It's essential for a lot of different things, component of chlorophyll, amino acids, plant hormones, nucleic acids, and so forth. If you don't have enough, you'll get yellowing of leaves and plants in the field and so forth, stunted growth. If you get too much, you can have problems, get problems with fruit development, just too much vegetative growth. Typically, we provide it in the media in the form of nitrate or ammonium, various forms of those. Plants can grow with just nitrate alone, but usually benefit from having some ammonium present also. But the other thing that's really critical is not just the total amount of inorganic nitrogen in the media, but that ratio of ammonium to nitrate can be really important as well. And I find that's fairly different between the different basal media that we're using. And we'll go through that a little bit more as we go through here. So the photo here just shows typical nitrogen deficiency symptoms in corn, the yellowing plants here, they're not getting enough nitrogen. But more important on this slide is just some of the common media just show that how much the nitrogen levels vary and that ammonium to nitrate ratio, how much that can vary in these different media. So MS that we've all used, that has one of the higher nitrogen contents, about 60 millimolar total inorganic nitrogen. The ammonium to nitrate ratio is about 1 to 2. If you go to the other end of that spectrum, woody plant medium, only 15 millimolar, so 4-fold lower inorganic nitrogen, but the ratio is the same, 1 to 2. Then intermediate, N6, B5, in that 30 millimolar range for total, but the ratio of nitrate to ammonium is very different in those, especially for B5, where it's much higher in nitrate. So that's good to keep in mind. A lot of whether it's good or bad for your species has to be determined empirically usually, but these basal media do differ a lot in their nitrogen content and in the ammonium to nitrate ratio. Okay, moving on. So potassium, that's the next most prevalent component in the media. It's essential for a lot of different functions, nitrate reduction, cell division, so forth. If you don't have enough, plants suffer, of course. It's usually provided as potassium nitrate in the media, typically in the 20 to 30 millimolar level. In the field, potassium deficiency is pretty common. This is just an example I picked out here in Kentucky in 2014. They're saying potassium deficiency was one of the key nutrient deficiencies in soybeans then, so obviously an important element. Phosphorus, another macronutrient needed for a lot of important functions. We're typically providing it by sodium phosphate in the media, or potassium phosphate, usually at the 1 to 3 millimolar level. This just shows purpling of corn seedlings that are not getting enough phosphorus under a field situation. Calcium, another one of the macronutrients, again, needed for a lot of different things, found in cell walls, helps regulate permeability. A lot of different enzymes are dependent on calcium. Come back to this, but the calcium cations aren't very mobile in the plant. They're needed in actively growing shoots. I'm going to come back to that in just a minute. It's usually provided as calcium chloride or calcium nitrate. One thing keep in mind, though, some people use calcium gluconate, and especially if you have problems with too much chloride in your media, some species are sensitive to too much chloride. Calcium gluconate can be a good alternative to that. And the gluconic acid also helps in the metabolism of calcium, such as something to keep in mind. Usually included at about 1 to 3 millimolar. This is an example I wanted to show that I think is interesting from the calcium perspective. I don't know how many of you have seen what looks like on the left here, these buggy whipping or necrotic seedlings. This is in corn. I've seen that a lot in some of the work that I've done in the past. That can be due to calcium deficiency. There's interactions in the media. If you have too much ammonium in there, that can cause problems with the calcium use and cause these type of symptoms. Now, it's very genotype specific as well. So in corn, B73 or stiff stalk inbreds are sensitive to this. Missouri 17 doesn't seem to be sensitive to it. But this was a nice study that was published just in 2020 that showed that at least in hydroponically grown seedlings, you can alleviate that by adding extra calcium and also by reducing the ammonium nitrate in the solution. So if you're seeing that type of a problem, you might be able to overcome it by just adjusting your calcium levels in the media. So it wraps out the macronutrients then. We got magnesium and sulfur. These are usually provided as magnesium sulfate in the media. Again, they're both in about the 1 to 3 millimolar level and essential for a number of different things, chlorophyll, ATP synthesis, and so forth. Let's move on to the micronutrient sense. The micronutrients, these are present at much lower levels, but are still important for plant growth. There's typically 10 elements that are considered as the micronutrients. Those are listed here. Over time, it's really taken better analytical methods and so forth to really understand which of these really are important. So better measurement capabilities come along to say, okay, yeah, this is an element that's important. Go through these. Well, actually, a question first. So these are very important elements for growth. Are they present in all of our media? Who thinks they're present in all of our basal media? Okay, yeah, they're not. They are, all 10 of these are found in MS, but you'll find that some media don't contain all 10 of these micronutrients. And for example, N6, woody plant media, DKW, they're missing one or more of these micronutrients. That means some of them may not actually be essential, but the other thing is they're needed in such small quantities that just trace amounts that be coming in from some of the other macronutrients or from the gelling agents or so forth are enough to provide what's needed. So let's run through a few of these. So iron is probably one of the most critical of all these micronutrients, key element in chlorophyll synthesis. Usually it's added along with a chelating agent such as EDTA, and there's really not a lot of variation in the iron levels in most of the common media that we use, typically included about 1 micromolar. Manganese, required for the activity of several different enzymes. Insufficiencies can result in chlorotic symptoms. Zinc, key element in chlorophyll synthesis. It's also used in the production of IAA. A lot of different enzymes require it. Moving through these. Boron. This is an interesting one. This is true of a lot of these micronutrients. There's kind of a fine line between having enough there to meet the needs and having too much to cause toxicity. And this is particularly true of boron. I found this quote that for many plant species, there's a very slight difference between the boron requirement and boron toxicity. And boron is actually used as an herbicide at times. So even though these elements are needed, you got to be careful not to get too much too. Some of them are toxic at fairly low levels still. Copper is an interesting one, so I'm going to spend a minute on this. So, I mean, again, it's used for a lot of different processes. It's essential, but only very small amounts are needed. High concentrations can be toxic again, usually provided by copper sulfate. It's not in N6 media. This is one of those that's not found in N6. The thing I find interesting about it, though, if you look at the range it's typically used, about 0.1 to 1 micromolar, MS media is at the very low end of that range, only 0.1 micromolar in MS. So there's been quite a few studies that have been published, especially in cereals, but also in some dicots, that raising the level of copper can actually help quite a bit in plant regeneration. So if you're having problems with plant regeneration, something to think about. Is my copper level too low? It might be, especially if you're using MS, because there's studies have found that raising it up to 2 to 50 micromolar or so can actually improve plant regeneration in a number of different species. And that does seem to be cultivar dependent, so even within wheat or some place where it helps, maybe some cultivars benefit more than others, but definitely one to keep in mind. And some of the other micronutrients, molybdenum, it's important in the conversion of nitrate to ammonia. Again, that's another one of those you don't find in N6 media. Cobalt, not considered essential, but it's a common micronutrient. It's not added in N6, woody plant media or DKW. Iodine, typically included in the media, it's not really considered essential. It's added through a potassium iodide. Then the last two, sodium chloride. Sodium's not considered to be essential. Plants will actively secrete it through the roots, and it's added just through a lot of the other chemicals we're adding to the media. So ferric sodium, EDTA, sodium molybdate, and so forth, it's coming in from a lot of other sources. Chlorine also is coming in from other sources, such as calcium chloride is a big addition of chlorine to the media, like I mentioned before. Some species can be sensitive to too much chloride ion, so you might think about using calcium gluconate instead of calcium chloride in those situations. Okay, that's all the mineral elements. Move on to carbohydrates. So obviously, we need a carbon source for the plants to grow in culture. Unless you have a photoautotrophic culture, there are some that can photosynthesize and produce their own carbon. That's not very easy to do, though. So most situations we're adding a carbon source. Jump on ahead. So the most common is sucrose. So you've got two of the disaccharides that are very commonly used. Sucrose is the most common. It's also the most common carbohydrate found in phloem sap. It's cheap, it's readily available. It's made up of glucose and fructose, so it's a disaccharide. But maltose is another common disaccharide used in culture. It works better for some species than sucrose. It's made up of 2 glucose subunits. And then the most common monosaccharide would be glucose. A lot of times it's added in addition to sucrose. It can be metabolized and used a little more quickly and readily. So the two together can be pretty useful. Fructose can be used. Now, fructose isn't used very often. Part of the reason for that is that it's more subject to problems during autoclaving. And this is something to think about with the sugars in general, that they can decompose during autoclaving, especially when they're present with the other mineral elements. So we spend all this time thinking about, the composition of our media, the exact concentrations of everything. Then we throw it into this pressure cooker, and things change there. So I think Ken's going to touch on that, the quality of media in his part of the talk too. But it's a really important thing to think about, is what's happening as you autoclave your media if you're doing that. Other carbohydrates that are commonly used are the sugar alcohols, now not typically as a carbon source, but especially mannitol and sorbitol. If you're using protoplast or particle bombardment where you need to regulate the osmotic pressure of the cells, typically we use mannitol and sorbitol to help do that. So those are very commonly used. Myo-inositol, usually about that more like a vitamin, about the 100 mg per liter level. Vitamins. So vitamins are also important. Plants grown in vitro can't make all the vitamins that they need for growth, so typically we're adding some other vitamins. The most common ones are the B vitamins. This shows the list of B vitamins that are commonly used, and the ones in green are those that are used most prevalently. I think in Piero's part of the talk, you mentioned that back White's medium originally used yeast extract in the medium, but it was found that yeast extract could actually be replaced by using thiamine, niacin, and pyridoxine. So as we learn more over time, we learn how you can have a more defined media and less of the undefined components that are added. But again, those green are generally added as vitamins. Some of the others are what I call in the kitchen sink media, especially for protoplast culture, things that might benefit from having additional vitamin sources present. So thiamine, one that's almost always added. Like I say, pretty much universally required for cell growth in plants. It's used typically in the range of 0.1 to 10 milligrams per liter. I think this is another interesting one because MS is pretty low in thiamine. It's got 0.1 mg per liter, the lower end of that range. And there's a lot of plants that can benefit from more thiamine. So most of the other media here, N6, woody plant media, and so forth, are using higher levels. So something to think about if you're using MS at that original low level of thiamine, you might be able to benefit from adding a little more. Nicotinic acid, commonly added, calcium pantothenate, a lot of time in each of these, pyridoxine, biotin, cyanocobalamin. So all of these are B vitamins that are fairly commonly added, again, not needed at very high levels. The only non-B vitamins used quite a bit in some species is ascorbic acid or vitamin C, especially in wheat and some other species. Added as an antioxidant can be useful in some situations. Amino acids. So plants grown in vitro, they can synthesize all the amino acids that they need. So amino acids aren't required to be added, but a lot of times they are added and are useful as supplements. They are good sources of ready-to-use organic nitrogen, and they can have other effects as well. So just a few examples of those that are commonly added that can have beneficial effects. Glutamine's one of the most common. Can be helpful in promoting embryogenesis when it's used typically in the 8 to 20 millimolar range. Asparagine, again, it's used fairly commonly, but usually at much lower levels, down to 100 mg per liter, about the 1 millimolar level or so. Again, it can be useful in promoting somatic embryogenesis, but it's also, there's some reports of inhibitory effects in some species as well, so it just has to be determined empirically. Glycine, often added, usually used at a pretty low concentration, 2 mg per liter or so. Proline's an interesting one, so that's helpful in promoting embryogenesis in a number of species. One of the more commonly added amino acids, also used as a cryoprotectant. When it's used, it can be used at fairly high concentrations, about 6 to 25 millimolar. I'm just going to use this as an example. Take a little bit of a sidestep here back from 40 years ago, my master's work. So that proline is actually one of the things that I've seen in my course of my career is having one of the largest effects I've seen in tissue culture media. So we're trying to induce friable embryogenic callus from corn. We're using the inbred A188. And what we found was that adding proline was one of the biggest things that helped in inducing these so-called type 2 cultures, friable embryogenic cultures of corn. Fortunately, we started with an N6 basal media, adding proline to N6 basal media went from no response at all up to fairly high levels of response. But the other interesting part of this is, like I say, we're fortunate we're starting with N6 media, so when we tried, if you do it in MS, you get no response at all. Adding proline to MS didn't help at all in getting this same type 2 culture response. Start playing with the nitrogen sources then, replace the N6 nitrogen with the nitrogen source in MS, you do see an improvement in response, or do it in the converse, take MS with N6 nitrogen. Yeah, so the MS basal salts with N6 nitrogen, you can see a response where you don't see it with just entirely MS. If you N6 with MS nitrogen, you take the response down versus just N6, but you still get a response. So the proline is really important, but it also is interacting with the nitrogen source, and a lot of other factors play a role as well. I think the main thing here is there's always a lot of interacting factors to think about in media, and I think Max is going to help tell us how to deal with a lot of those interactions, and you've got so many things to think about, how you can use machine learning to help in some of the media optimization. Okay, undefined organic supplements. So there's a lot of times we're adding things that really just aren't that well defined to media, and they can help. So there's a reason we add it, because we try it and they make a difference. Some of those things like yeast extract that we've already talked about, protein hydrolysates, a lot of times we're adding case amino acids to our media, powdered fruits and vegetables, coconut milk, coconut water, tomato juice. There's a lot of different things that have been added to media over time and that have promotive effects. A lot of times we don't really understand what is it exactly in there that's having those effects, but if it's not broken, don't fix it if it works. But it's also part of what's given tissue culture more of the more art than science type connotation with some people in too. So over time, we've learned more about what it is that's important in some of these elements, and we're able to replace those with more defined ingredients, but in some cases we still don't fully know. So case amino acids is one of the more common ones of these, relatively undefined ingredients. That's basically just protein from cow's milk. It's got a mixture of 18 amino acids in it, but it's also a number of other things, calcium, phosphate, microelements, and so forth, that might be having a beneficial effect. There have been studies done just replacing the amino acids. They can help, but there's usually more to it than just that. So it's not just those amino acids that's giving the promotive effect. Activated charcoal is often used. In this case, it's not a carbon source for growth, but it's just having other beneficial effects to some species, but not to others. One of these things has to be determined empirically. Probably it's binding up other things in the media in that case. Activated charcoal is just charcoal that's been activated by exposure to high heat. Yeast extract we talked about already. In addition to the vitamins we talked about, it also has free glutamic acid. Malt extract has been used. It's a co-extract of corn and malted barley, sometimes helps in somatic embryogenesis. Coconut water has been used a lot in the past. I don't know if you know there's a difference between coconut water and coconut milk. So coconut water, it's if you drill into the coconut and drink it, you're getting the coconut water, the endosperm. Coconut milk, that's the grated meat of a mature coconut. So there's a difference to keep in mind there. Coconut water has a lot of different things in it. I always thought of it as really just having more of a cytokinin effect, but there's a lot of other things going on there. It does contain diphenylurea, which has cytokinin-like activity, but it's got proteins, amino acids, vitamins, minerals, a whole bunch of things that could be contributing, and I don't think we fully understand in a given system just exactly what the promotive effect is, but if it works, why not use it? So last but not least, just briefly go through some plant growth regulators, and then we'll turn it over to Ken after that. He'll be talking about the solidifying agents and quality control aspects of media. So everybody knows about plant growth regulators. They can be classified in different ways. One way to do it is into auxin, cytokinins, gibberellins, abscisic acid, ethylene, and there are a few others you can throw in here. Auxins typically trigger cell enlargement, root growth, important for somatic embryogenesis. Cytokinins trigger cell division. They're important for the growth of shoots and overcoming apical dominance. Gibberellins typically involved in stem elongation. Abscisic acid promotes seed dormancy, often used for maturation of somatic embryos. Ethylene is the only gas out of all these, and it's involved in fruit ripening, organ excision, and so forth. Not all these interact, of course, at the molecular level, and sort of the classic cytokine and auxin balance for root shoot growth, of course, we're all familiar with. But just real briefly go through a few of the most commonly used ones in each of these classes. So auxins, IAA, it's the original auxin isolated in 1937. It's the only naturally occurring auxin. The reason probably isn't used even more than it is that it's one of the least active of the auxins, and it's also very sensitive to degradation. It's light sensitive, it degrades very easily in general. So people generally tend to use the synthetic auxins, so IBA is very commonly used, especially for rooting. NAA is the most commonly used auxin. Then, especially in cereals, but also some other dicots as well, we've got the auxinic herbicides. So things like 2,4-D, dicamba, picloram, probably very active, usually at lower concentrations than the IAA, IBA, and NAA, and used in a lot of cereal species. There are a number of others as well, PCPA, 2,4,5-T, chloramben, other auxinic herbicides that are also sometimes used. Cytokinins. So kinetins, top of this class. The first cytokinin isolated by Skoog back in 1947. BA is the most widely used cytokinin, though. It's a synthetic cytokinin. 2IP, zeatin, metatopolin. Zeatin, the one thing kind of holding back more widespread use of that probably is the cost. It's inexpensive, and it's also has somewhat lower activity. So you need higher levels, and it's also fairly expensive. Metatopolin's relatively new. It can be used at higher concentrations generally than BA without inhibiting rooting, so that's been one of the reasons people have been interested in using that. Might be seeing more use of that in the future. TDZ is an interesting one. So TDZ is an activated substituted phenylurea. It actually has been reported to have functions both similar to cytokinins and auxins. Now, it does inhibit cytokinin oxidase, so I always kind of think of it as, kind of cytokinin effects. It's increasing the lifetime of cytokinins in the plant, but it does have these other auxin-like effects in some species, so it's kind of an unusual one. This quote out of a fairly recent paper that despite decades of research and widespread commercial application, the specific modes of action of TDZ and plant growth regulation remain undefined. So a lot still kind of unknown about its action. Get to some of the other plant growth regulators. Gibberellic acid, often used to help with stem elongation. The flip side of that, ensimidol, blocks the synthesis of gibberellin, so that can be used to reduce the internode length. And a really nice example of that, I think, is the recent work of Corteva, where they're doing seedling transformation in corn, so to keep the plants shorter in culture, which helps use those, just add ensimidol, and it keeps them nice and short and easier to work with, and hasn't had any detrimental effect. I think there's a positive effect they reported on culture response and transformation. ABA used for somatic embryo development in a lot of cases. Ethylene, if you want to manipulate ethylene in cultures, there's usually two ways that that's done. You can add ACC, which is then converted to ethylene by ACC oxidase. So if you want to increase ethylene, ACC is a good way to do that. More often, people are wanting to control the action of ethylene, so silver nitrate or silver thiosulfate are added because the silver ion blocks the action of ethylene. That's another one of those, because after proline, that's probably one of the other biggest effects I've ever seen in the course of my career, is the effect of silver nitrate in culture. And again, this goes back to B73 cultures we're trying to initiate, and some work that Dave Songstad did. Night and day difference between no silver nitrate. If you had silver nitrate in the media, just much better culture response, presumably from controlling the action of ethylene. So in some systems, it can have a huge effect. Then just included a few references here. There's a lot more that can be learned about all these, of course. Some of the companies like Phytotech, Sigma, Duchefa, they have really good information on their websites on all the media components. Some good review articles here too. The one by Greg Phillips at the top is relatively recent, nice review article. So I'll read it back to those for more information. And I think we'll turn it over to Ken.
What we're going to do now, we're going to open the floor for questions, if we have questions. And there is a microphone there, so if you want, you can walk to the microphone, or I can walk over and get the question, and then we're going to answer them on the floor by the panel. So if you have a question, come to the microphone, or I can, if you raise your hand, I can come over and have you. Okay. Joyce?
Thanks for the informative presentation, Chuck. A lot to cover there, experience-wise. So I was curious, you mentioned proline made that big difference between getting embryogenic callus. What made you decide to add it? Do you know the mode of action? What caused that reaction?
Sure. So in terms of why did we look at it, first off, the main reason there was studies have been done by Bill Sheridan looking at immature corn embryos and the composition, and proline was one of the, at very high levels in immature corn embryos, and so that's what triggered us to look at it in the first place. in terms of what it's doing after 40 years, I don't know. I mean, we think it could be a reduced nitrogen source. It's probably involved in stress responses somehow, but we really don't know for sure what it's doing.
Okay, next question.
So I have a question. When you're troubleshooting for your culture and their response, what would, with the different components of the medium, what was the first thing that you were looking at first? Like the thought process as to this is where you're gonna, when you're optimizing and stuff.
Yeah, good question. It's just like on a new species or something, where to start optimizing, is that the question? Yeah, so I don't know, it'd be interesting to hear some other thoughts too, but I think you might start, like some of these basal media are very different, right? MS is very different from woody plant medium, is very different from B5. You can start with some defined media, just look at the initial response there. Of course, the typical auxin cytokinin grid is always a good place to start. But and then once you start seeing a decent response, then again optimizing from there. But Max might have better comments too, so you'll be getting into that in your talk. But okay, okay. Thought that Ken will be addressing that in his talk too.
Next question. Okay. I think I mentioned in my introduction, all the material is going to be available if you register through the on-demand program, and then the recording of the session is going to be posted on the Society YouTube channel, so you can review all the material. And even though I didn't ask the panelists, I'm assuming that if you have questions that they come after you watch the video, you can contact any of them and they can answer your questions. I also forgot to mention that we're going to have a coffee break at 1:40-ish. That is going to be, I believe, right behind these doors, but I will confirm as soon as I can. But definitely we're going to have a break around 1:40, 1:50. We're now going to Ken presentation about media preparation and quality. And then around maybe 1:50, I'll give you kind of the time point. Okay, thank you.
So I want to thank the Society, Piero, and Michelle for the invitation to come here. It's been a while since I spoke, so I'll try not to embarrass myself up here. And if my southern accent becomes too much, just raise your hand and I'll try to pronounce what I was trying to say. As Piero indicated, I guess I'm the old guy up here. So, and as I look at all the young faces, I'm kind of like astonished, but I'm really glad to see everybody here. I attended my first society meeting in 1982, so it's been a few years, and this has probably been one of the best scientific societies that I've ever participated in. So I encourage you guys to get as many of your colleagues to come to these meetings and everything. I think there's a lot of information that you can gain. When Michelle approached me about doing this talk, see if I can get this. When Michelle approached me about doing this talk, it kind of hit home with me a little bit, because as I used to visit customers in research labs, micropropagation labs, when I was either at Sigma or Phytotech, one of the things that I kept getting quite a bit was, how do you optimize a media? You know, people just didn't understand what the next steps were. And I realized that the companies that have developed the prepared media have made it much easier for you to do your lab work, but at the same time, or on the flip side, it has, I think, caused a lot of information to be lost as to how you typically try to approach these. For those of you that don't know me, I began working in tissue culture in the mid-'70s. I was very fortunate in 1977 to go to the W. Alton Jones Cell Science Center and take a course on plant tissue culture under Dr. Murashige and a few others. And it was really an eye-opening opportunity for me to be able to sit down and talk with Murashige about how he had designed the experiments and everything that he went through to come up with the Mirshig and Skoog media. We talked about all the factorial experiments that he created and how much time it took in terms of optimizing the media. Since that time, I've tried to work on optimizing media throughout my career using single, double, and multiple factorial experiments. Now, Max may blow this out of the water, as there's much better ways, I'm sure, now to do this, but again, I'm old school. From the time I was at LSU, we developed an orchid media there that has become one of the most popular orchid media that's used today. And after my time at LSU, I went to work for Sigma. We started the first plant lab that was dedicated to manufacturing media for plant tissue culture. After leaving Sigma, I spent a few years at a small biotech company in the Kansas City area that primarily manufactured mammalian media, and there I worked on developing large liquid delivery systems that would deliver anywhere from 100 liters up to 500 liters of media at a time. Then after that, I did start Phytotech Labs and was there for 21 years before deciding to retire. So over the last, many years, I have seen a lot of progress that has been made in media development. As I said, Max is going to have a great presentation at the end of this talking about experimental designs, and hopefully, I know a lot has changed over 50 years, and hopefully he'll be able to bring some of that to your attention. So before I get into the meat of this, I would like to ask a few questions and just kind of poll the audience. First of all, how many of you are actually involved in research of plant tissue culture at a university, biotech company, commercial laboratory? How many of you are actually involved in the day-to-day research? Okay. Now, how many of you work at a micropropagation lab where you're commercially mass-producing plants? I see a couple. Good. That's great. And then now, how many of you in this room are actually responsible for making the media that is used in your lab? Okay, better than I thought. Okay. Again, one of the things that I did run across a lot when I was visiting customers and talking with some of the lab managers is they would tend to put the chore of making media off on someone else, and those people were the ones that really didn't understand, why you use the media. They just said, oh, I just go grab a bottle and I put it in the water. Well, there's a lot more to the media than just that, and that's what I hope between this year and next year, some of that will address. So again, Chuck has talked about some of the major nutrients that are used in the plant tissue culture media. I want to briefly talk about some of the solidifying agents. Solidifying agents aren't necessarily critical. You can do tissue culture without a gelling agent by using filter paper bridges, cellophane type wicks, polyurethane foam, and things like this. So gelling agents have been used since back into the 40s when Philip White started doing some of the work, and some of the later people, they were using gelling agents, and at that time they primarily used agar. Now, one of the things that I'm going to try to hit home about today is the quality of the raw materials that you're looking at. It is very important that you know your source of gelling agents as well as many of your other raw materials. It becomes very, very important as you move into trying to get good replication of your research or your production if you're in a commercial labs. One of the main advantages of using a gelling agent is that it provides a physical support for the plant tissue culture or the plant culture to sit on so it doesn't get completely submerged in a liquid solution. There are some disadvantages, though, of using some of the gelling agents. First of all, you can have impurities in the agar, which I'll talk about in a few minutes. They can also, it's believed that they bind up with some of the nutrients and thus take some of the nutrients away from the plant's ability to be able to use those nutrients. You have hyperhydricity, which is, of course, the watering, the glassy look of the plant tissue that you have that is prevalent with certain gelling agents. And you have issues with the gel setting properly. Again, we'll talk about some of these more in detail. And so the only way of really getting around some of these disadvantages is to go to more of an all-liquid system, such as a bioreactor. Max showed me some photos earlier of things that he's developed in his lab that look very promising in terms of growing cultures. So I'm going to first start with agar. So agar is basically developed or grown as seaweed or kelp, which many of you may know. It's typically used at concentrations anywhere from 6 to 8 grams per liter, but for a firmer gel, you can go up as high as 12 to 15 grams per liter. It is required to have the proper pH to get a proper gel. Again, one of the questions that we used to get a whole lot at both Sigma and at Phytotech was people would call up and say, my agars are not gelling. First question we would ask, when did you adjust your pH? pH is very critical when you're using all of these gelling agents. Now, agar is pretty good in terms of shelf stability, particularly up through the first 5 years. You really don't have much issue with shelf life of agar, but after that, you will begin to get some breakdown, particularly between about 8, 10, 15 years in the mechanical structure of the agar. So my recommendation is that, try to use it up within about a 5-year period. Gel strength can vary quite dramatically from one lot of agar to the next. The thing that you need to think about when you're thinking about agar is that this is a biological product. Okay, it will vary from lot to lot, from year to year. It will vary depending upon where it's harvested, whether it's the Mediterranean, the South Atlantic, the Pacific. All of these have a role and play a role in the quality of the agar that you get. That's why I strongly recommend people to get a sample of agar before they're starting. Do a test, look at the gel strength of it, look at the purity of it, before you move on. And this is especially important if you're switching vendors. And I'll talk a little bit about vendors later on, but it's very important that you know the quality of your agar, because it can have a pretty dramatic effect on the results that you're getting depending upon the source. As I said earlier, pH is critical. You need to make certain that you have a pH of at least 5.2 for it to properly gel. Again, that is a common thing that we would run into, is that people would adjust their pH before adding the gelling agent. And as a result, because of the variations of the impurities in the agar, that might affect the pH, and it would end up with some gels not gelling properly. So again, I know in the past, or a long time ago, they used to say always adjust your pH before you add your gelling agent, and I've been preaching on this for a long time. Make certain you adjust your pH after you add your gelling agent, and then just make sure you rinse your pH probe out completely. So agar is primarily derived from 2 species, Gracilaria and Giletium. And again, a long time ago, about all that was available was Giletium agar. But over the last 20, 30 years, Gracilaria has become available. Gracilaria has a stronger gel strength. It can usually be used at a little bit lower concentrations. And so it has the advantage also that it produces a little bit clearer gel. Now, Giletium is typically a softer gel, so if you've got a tissue where you're wanting to get possibly better root development or study root growth, then the Giletium agar is one that you might want to consider. Again, it's not as clear of a gel. It does not produce as clear of a gel, and sometimes has to be used at a little bit higher concentration, like 8 to 12 grams per liter. Giletium is available in both a purified grade and a regular grade. The purified grade, basically the only difference is that a lot of agar has been washed 2 or 3 times more during the processing, and that results in it being slightly pure. For most research, you don't necessarily have to use the purified grade. There are some species, though, that you will find that the purified grade works much better. And again, because this is a biological product, if you're working with multiple species or even multiple cultivars in your lab, you may find that one source or one type of a gelling agent will work far better on one cultivar species than on others. So two other gelling agents that are also used, carrageenan, which has been around for a long time, but it is typically the kappa type of carrageenan that is used in plant tissue culture. It produces a fairly strong gel, but it is used anywhere in concentrations of 6 to 10 grams per liter. Gelling gum is a process of bacterial, a process during a bacterial growth, and is harvested from a bioreactor that is growing large vats or volumes of bacteria. It has a little bit softer gel strength, but it produces a very clear gel, which I'll show you a photograph here in just a few minutes. It can usually be used at 2 to 4 grams per liter. The other thing with both gelling gum and agar is that most of the companies out there now produce a blend of these. These will run with about 1 to 1.5 grams of gelling gum and somewhere between 4 to 6 grams of agar. Again, different companies have different formulations, so you need to kind of look at them and see which one is going to be best for your particular applications. Now, another thing about gelling gums is that it does require the presence of calcium and magnesium ions for it to gel properly. When this first came out, I'm going to say in the 80s, people started trying this, and one of the comments that I would get at Sigma is, well, it's not gelling. And my first question then would be, what media are you using? And quite often the people that had problems with it gelling was using a media that had very low concentrations of calcium or magnesium ions in it. So it's important that you do understand that if you're going to use gelling gums, that you need to make certain that you have the presence of these calcium and magnesium ions in it. This chart here basically shows the optimal concentrations of the calcium and magnesium ions to get proper gelling. Gelling gum is also, it is critical that the pH on it is at least 5.2 or above, or it will not properly gel. Now, you heard me a few seconds ago use the word gel right. When gelling gums first came out, it was manufactured by a company called CP Kelco, and they trademarked the name as gel right. So I will sometimes throw that out just because it was around when I was more in the lab and everything. CP Kelco no longer makes gel right. They make a product called Gelzan, which has replaced it. But you can also get other grades of gelling gum from other sources. The thing that you do have to be careful with, again, is looking at the quality and the manufacturer that you're getting this from, because it will vary from lot to lot. This next slide basically just kind of shows you a guide in terms of clarity of the product. And as you can see, on the far left side is the gelling gum, and on the far right side is agar. And this is basically just kind of a flow chart down as to what gives you the highest clarity. This next slide maybe does a little bit better job of it. Again, you can see the different grades of agar on the right-hand side, and as it gets clearer, you're going toward the far left with the Gelzan. So next I wanted to kind of, let me go back to one thing before we go back to this. And this is, again, I can't stress enough how important the quality of your raw material is in any plant tissue culture work, okay? You have to realize that agars in particular, and as well as carrageenan and gelling gums, are commodity products. Okay, they're used in the food industry. The food industry takes the largest volume of these, and that can dictate price. The environmental conditions in which the agar is growing in the ocean has a dramatic effect on the quality. These climate conditions, environmental conditions can vary from year to year. Some years the harvest of agar is very slim, and other years there's an abundance of agar. And as you can imagine, that is going to alter the price of the product from one year to the next. So you do need to think about when you are choosing which gelling agent is going to work for you, kind of keep these things in mind in terms of how is that going to affect my research down the line and stuff like this. And that kind of leads then into my part of my next talk, where I talk about the quality of raw materials. So when I first started in tissue culture, there wasn't, basically you just had a reagent grade chemical that you could get. They didn't really have a lot of sources for ACS or USP grade chemicals back then. Some of the stuff I think honestly probably was agricultural or industrial type grade. Again, this goes back to the quality of the products, particularly on things like ammonium nitrate, potassium nitrate, that are primary components of fertilizers. You need to make sure that you are getting a good quality reagent or ACS grade material for your research. If you're not familiar, I think most of you probably are, but ACS and USP EP, these are different grades of chemicals that must meet certain specifications, okay? They are outlined, they're very definitive, and for a chemical to meet that particular grade, it has to pass all of these tests to be labeled as that. You will sometimes see chemicals sold as a combination. It says ACS, USP, EP grade. That's probably going to be your best choice. That's going to be the purest material. USP stands for US Pharmacopoeia, EP stands for European Pharmacopoeia, and there are slight differences in the requirements for each of these depending upon the pharmacopoeia, but as a general rule, these are your better grades to work with. Now, if you're from outside the US, you probably will not be able to find ACS grade materials. You probably won't be able to find USP grade materials, but you should be able to find either EP grade materials. There's also a BP grade, which is British Pharmacopoeia, and there is a Chinese grade, and I think it's just called the CP grade, which is a pharmacopoeia that was developed in China. So the reason that I stress so much about looking at the quality of your raw materials is the lower quality is going to have more impurities, okay? And if you were trying to develop and figure out why your tissue is responding or not responding, it sometimes can very well just be simply the impurities that are in the chemicals that you're buying. Now, most of the companies out there that are providing prepared tissue culture media, they, for the most part, I believe, use ACS, USP, or EP grade chemicals, and they have them for sale as well. But there are other companies that sell media out there that do not have these grades of chemicals. So that is why it's important, and I kind of keep stressing on this a lot. The other part of this is the verification of quality. Okay. For the most part, if you know your vendor and you've worked with them for some time, you can simply rely upon their certificate of analysis if you trust their material. And that's what I hope for most of you, that you've got good vendor relationships and it is a situation where you've never had any problems with any of their chemicals. However, if you're getting into what I'm going to call a bulk operation, or you're going to have to really scale up what you're doing, and you want to go to a source other than some of the companies that are out there currently, it's important that you begin to verify the vendor that you're getting the material from. This can include like an on-site visit of the manufacturer. It can include basically you doing identity and physiochemical tests within your lab, or whether you do it within your lab or whether you contract it out to another lab or another company. You need to always make certain that you were checking the quality of these products. Some of the things that we typically would look for is we would do an identity and purity test, and then we would always look at physical appearance, solubility, pH, osmolality. We'd run FTIR or IR analysis for identity, GCHPLC analysis for purity, and in some cases we'd have to run mass spec analysis just to make sure that we were getting what we were wanting. Now, you might go, why is he harping so much on this? I'll give you a couple of examples that I have run across in my career where this was a problem. There was one company in which we bought between 1,000 and 2,000 kilos of potassium nitrate from, and we had actually obtained a sample in advance from the company, tested it, everything passed. However, whenever that material came in, while it still passed and met USP specifications, there were actually black spots all throughout the potassium nitrate. That material basically we could never use. Okay, if you used it for manufacturing a prepared media, you would then have been selling something that would have a precipitant in it because those black spots did not go into solution. You couldn't use it to make a liquid media because that would then end up clogging your filters as you were filter sterilizing the media. So, to say the least, we never used that vendor again. But, it costs money whenever you run into situations like this. So that's one of the reasons that I really stress so much that you look at the quality of the vendor that you're getting materials from. Another example is we had bought a cytokinin, again it was a new company. We pre-evaluated them, we pre-tested a sample lot. When we got the bulk material in, we just knew something was not right. It was not performing in biological tests that we were running the way that we expected it to. When we actually then did a mass spec, I believe it was mass spec sample of it, we found out that it was a mixture. Instead of being all trans, it was a mixture of cis and trans, and the purity was nowhere near what the sample was that they had sent to us. So I'm trying to, educate you on these things so that you can learn from our mistakes, if you will, and know what not to do. So the next one again, and I have kind of hit on most of this already, is to know your vendors, build a good established relationship with them, and make certain that you're getting the best quality of products, because just little things can throw off your results. Now, one of the things that I recommend for most companies, if you're doing particularly critical research, is to have a primary vendor and a secondary vendor. Okay, your primary vendor can be your main supply of your powdered media, growth regulators, gelling agents, stuff like this. But it's always a good idea to have a secondary vendor that you can rely on, that you've tested their products, and that you're comfortable with the quality of products that they're producing. If you're doing anything that is potentially going to be pharmaceutical related, this is a very key part of that, because the FDA and other organizations that regulate pharmaceuticals are going to require that your manufacturing people have multiple vendors in case there is an issue. Now, one of the things that I know we used to do, and I think most companies, I guess, still do this, is that if you sit down in the beginning and you know what your research is going to be, you know calculate roughly how much media that you're going to need. My recommendation is that you get all of the same lot of either that prepared media, plant growth regulator. Figure out what you're going to need to get your entire research or your entire production cycle, if you're producing micropropagating plants, through with one lot of material. Now, if you don't have the storage space for, let's say, holding 5,000 liters of media or something like that, I believe most of these companies still will ship it to you on a monthly basis, the same lot. They'll hold it and keep it under the proper storage conditions and get it to you when you need it. One of the things that I also recommend is that people create a receipt of materials. Again, something that I did see quite a bit as I visited some customers is they would go and pull a bottle off the shelf and say, oh, this is what I use. How old is it? I don't know. When did you receive it? I don't know. In some cases, they couldn't even read the writing on it as to where it came from. It's important that you keep a log of all the raw materials that you bring in. This can be a digital log in a spreadsheet. It can be a log that you keep on paper. But record down when you receive the material, what the expected shelf life of the material is, what the storage conditions are, when it should be retested, and of course the vendor source and everything. This will help you down the road if you do begin to get some questions. You can always go back to your receipt log and figure out where it came from. One of the other things that I have been a strong advocate of is the use of SOPs. How many of you in here have SOPs in your lab currently? Okay, that's a definite increase from what I used to see a while back, but there were still a lot of people that didn't raise their hands. SOPs are very critical in terms of outlining exactly what steps are supposed to be done in the lab and how they're going to be done. Again, as I visited some lab, they would just be writing things down on a notebook if they even did that. Or one person might make it one way and another person might make it another way. These are all little things that you can do to help improve the quality of your research and of your production. It should include written instructions of weighing chemicals, taking pH solutions, calibrating the balances and pH meters, how to make stock solutions, how to store things. I mean, anything that you can think of that might affect the quality of your research or your production, you ought to have it into an SOP. This is just a very basic SOP form. I was actually surprised as I was pulling some of this together. Places like Google Docs, Microsoft Word, they all actually now have forms or examples of SOPs that you can use to create yours if you don't have them in your lab. There's 5 major components to an SOP. One is the purpose. Again, that's just stating how to make a stock solution, okay? That's the purpose. Procedures, a detailed step-by-step procedures, that's what needs to be done. Think of this as like baking a cake. Whenever you go to bake a cake or make some cookies, well, unless you use it from pre-mixed, you generally have to add things in a specific order. The same thing should apply for this as well. You need to make certain that in developing your SOPs, you have it very specific as to how things are going to be laid out. Your scope, this is the limits and boundaries that you kind of want to follow. At this point, you want to make certain that, in a lot of cases, the scope and purpose are lumped together. Responsibilities, this is who is going to be actually doing the work versus the approval signatures, and then the title pages. That's, you know, basically what you're kind of looking at now. It would be a sample of a title page. One thing that I want to stress is always have addendums that contains the forms that need to be used, whether they're electronic or paper. Make sure that you have that completely spelled out. Okay, at this point, it's kind of a good stopping point. I'm going to take a break, and I think the coffee, I see some right out here, and then we can pick up in just, what is it, 15 minutes?
Yeah, I would say let's come back.
So I had a few very good questions during the break, and if I forget to address them, please don't hesitate to stand back up and re-ask the question. So I want to continue on in terms of media preparation. Again, this chart basically just shows all of the components of a media. And we've talked about everything except for water. I would hope in this day and age, most people are using a very polished DI-RO water, at least double distilled water for your lab. Again, water quality is going to be just as important as any of your nutrients in terms of what type of effects and results that you get. Now, there is one thing that I want to point out about water. Again, assuming that most everybody is using either DI or DI-RO water, is take into consideration the environmental conditions around your lab, okay? And what I mean by that is a lot of times the pH of your water can vary depending upon the time of the year. It can vary depending upon whether or not you've had a lot of rain in the days preceding you taking the water. I know you may kind of think that sounds crazy, but I have seen it multiple times where a person has had their lab, they had tremendous amount of rain a couple of days before, and their pH is off, and it is in part due to the pH of the water being off. One of the things that I recommend that you do as part of your laboratory protocols is that you take the pH of both your tap water and your processed water at least once a week and record it down. And that way, if you do run into some issues, you can maybe be able to go back and attribute it to heavy rains or something that's occurred in your environment. I'm going to go through this very quickly because Chuck has already talked about this. And this is terms of nomenclature. When we talk about a basal salt mixture, this is basically the inorganic salts and that's it. It does not contain any of the plant growth regulators, things along this line. A basal medium is any medium that contains at least one of the other components, such as carbohydrates, things like that. Plant growth regulators, again, these can be both natural and synthetic growth regulators. Hormones are generally what we would be considering things that can be naturally found in the plants. Macronutrients primarily are your nitrates, ammonia, and potassium and phosphorus. These are typically at concentrations higher than 5 millimolar. Mesonutrients are the other nutrients that are typically between 1 and 5 micromolar, micronutrients less than 1 millimolar. So I want to talk a little bit about media preparation and how it's worked. And again, part of this conference was being put together in part because there was a general consensus that people were starting to lose some of the basic information on how to prepare media, how to formulate media, if you had to optimize a media, what you needed to do. We've already discussed up to this point all of the primary components of the media. Now I kind of want to talk about some of the options that you have available out there. The pre-made media that's available, I know, has made it very convenient for the labs and everything. One of the advantages of using pre-made media is it eliminates potential mistakes by technicians in making media. It offers greater consistency in your media from one production to the next. And when you take into consideration labor, overhead, things like this, generally prepared media is much cheaper. Now, the disadvantages of using prepared media is that we fall into this trap of just pulling the bottle off the shelf, putting it in, and not really thinking about, the steps and what we're doing. One of the things that I found when I started working at Sigma and I was working with some of the commercial labs, they were very hesitant to try using pre-mixed media. There had been some issues in the 70s with some of the media that had been produced and everything, and they had a reasonable argument for being skeptical. However, once I convinced them to begin looking at this and trying this, after a couple of years, nearly every lab that we worked with would come back to us and make a comment about how they had seen such an improvement in the consistency of the media they use for mass-producing plants. And it all boiled down to little errors that were being made by people that were making the media that you just wouldn't even think about that was causing some of the problems. Now, again, if you're using a premixed media, it becomes a little bit more difficult to try to optimize your formulation. I'm going to talk about that toward the end as ways of getting around it. You can also, like we did back in the 70s and before, you can make your media from stock solutions, okay? Stock solutions have the advantage in that it's much easier to adjust the volumes of a particular element that you're going to put into it, and that makes it a little bit easier to manipulate what you're trying to do. Now, one advantage with stock solutions is that I think that there are several companies out there that now make stock solutions of these components. They may have 2 or 3 items or 2 or 3 components in the stock solution, but that does make it a little bit easier, again, in terms of lowering or raising the concentrations of particular elements to try to determine what is going to be best for your plant cell growth. The other advantage of a stock solution is that if you buy it from a company, they are typically making batches that are anywhere from, let's say, 100 to 500 to 1,000 liters at a time. Again, so it goes back to the consistency that you're getting. You're more apt to be able to get much greater consistency over multiple passages of your growth when you're not having to make the stock solutions fresh every month or every 2 or 3 months. Now, I want to talk a little bit about how media preparation is done. Now, prepared media has been available since the 60s with mammalian cell culture, and the first prepared media for plants came out in the early 70s. So this information is not new. It's been around for over 50 years. The technology has been there for 50 years. It's proven, and it does work. Now, I can't get into too much detail about how this is done because of proprietary reasons, but basically what you see here is, in this shot, is a ball mill, and basically the components are added into this ball mill. There is a set sequence in which you have to add the components. There's set times that you have to follow. This should all be laid out in the SOPs. You can't just take everything and dump it into a mill at one time and expect it to come out. It will not, trust me. So we talk about tissue culture being an art and science. Making media is also an art and science. We try to develop very stringent SOPs that outlined each step, how long things needed to be in there, what your milling cycle was, so that we had consistency of products. If you leave it in the mill too long, you end up with a heat buildup that can end up in degradation of the chemicals, cause precipitation, can actually begin to cause the chemicals to just bind together and form what we used to call big marshmallows, what it would look like inside the mill. So this is, again, it's proprietary, but most of these companies have done a pretty good job of figuring this out. Now, that being said, every company, because of proprietary issues, probably has their own way of doing it, okay? And that will end up resulting in variations of quality of the media from one company to the next. Some of you may have experienced some of that. Lost my train of thought there. Anyway, I know what I was going to say is, as I said, most companies have developed pretty rigid protocols for doing this, but I will tell you that in my career, I have run across companies that are selling media, none of them in the US, but companies that are making media that basically we're doing it in stainless steel drums, or we're doing it literally in bathtubs. That's not the way to buy the media. You're going to find a lot of inconsistencies and stuff like that. Again, the companies in the U.S. Have really developed very good protocols for doing this. Now, as I said, ball milling has been around for 50 to 60 years. There is another technology. Well, let me go forward one. Ball mills come in various sizes. I mean, if you're really ambitious and wanted to do this, you could probably get into making media on yourself for $5,000 to $10,000. But again, you've got to have very stringent protocols and figure out what you're going to do to make it work. These are what we would consider to be laboratory grade manufacturing mills that you can use and make batches as low as 100 liters at a time using some of the smaller mills. Or you can use some of the bigger mills like you saw in the previous photograph that end up making quantities of 50,000 plus liters at a time. The process, as you saw in the previous picture, was the materials went into a drum. You add a grinding media, which is basically zirconia grinding stones. And again, these will grind the materials into a very fine powder. If you don't have the right timing, you can end up with, pieces that are not ground properly and they're non-homogeneous. Or if you, again, grind it too long, you can have other problems. Again, you are not going to be able to find this information, much information from some of these manufacturers, but use a good quality manufacturer, test their products, and make sure you're happy with their results. Now, there is another way in which media can be prepared, and this is called jet milling. This is a process that is primarily used in mammalian cell culture now. At this time, it's, I think, only been around for about 15 or 20 years. But in this process, basically, the raw materials are input through metered dispensers into a somewhat round chamber, and around the outside of that chamber, air ports are going in, and the high pressure air basically breaks all of the components down, and then it goes through an output where you then collect the media. One of the advantages of jet milling versus ball milling is that you theoretically can have a continuous production of powdered media, so you can make bigger lots. Some of the companies that I have worked for had moved to this, and within the mammalian industry, sometimes that you are getting companies that need literally 500,000 liter lot sizes of media, and this is really about the only way of doing it is through jet milling, because you are limited with the size of the ball mills. So again, with stock solutions, it's very critical. I can't keep harping on this enough to have your SOPs to really outline it. And one of the things that you need to think about, and I think that is so important, if you have very good SOPs, if a technician leaves or you hire someone new, it's much easier to sit down and hand them the SOPs and say, all right, sit down, read these, study these, learn these, and then everybody follows the same procedures. Again, in making your stock solutions, you can have it for your inorganic salts, you can have it for your vitamins, your plant growth regulators. One of the things that I do recommend is you have to do a little bit of research and figure out what is the best sequence of adding things. And just like with powdered medias, you can't throw everything in at one time and mix it up and expect it to work. A lot of times you'll get a chemical reaction and things will precipitate out. The other thing is that when you're making particularly things like plant growth regulators or some of the vitamins that are not water soluble and you're having to use a solvent, you need to make certain that you use the solvent, the same solvent every time, okay? Again, labs that did not have good record keeping or did not do this would sometimes use hydrochloric acid one time, sodium hydroxide another time, something else. Another time is they made stock solutions. That's only going to lead to inconsistencies in your results as you go through. So it's very critical that you pick one solvent to use and stick with that through your entire sequence. Now, the other thing that I want to kind of also point out, Chuck showed a picture at the very beginning of people mouth pipetting things, okay, which, yes, I know that was done a long time ago. But the other thing that kind of goes along with that is when you get to your stock solutions, again, I go back to try to figure out how much media you're going to need to complete your research, okay? Then make certain that you make your stock solutions in large enough volumes to do all of that research. So you're starting with one batch. Now, you may not be able to store, or you may not have a container large enough to store that in. That's where you would begin to break it down into aliquots and then store the small aliquots and then pull them out on a weekly or monthly basis depending upon what you're using. And it also, the mouth pipetting goes back to a thought that I had as well that I used to see and run into a lot, is if you've got multiple people in the lab using the same stock solutions, I highly recommend that, again, you break it into aliquots and each person gets assigned their own aliquot. That way you don't run into issues where people are sticking a pipette in 2,4-D, then using the same pipette to stick it in a vitamin solution or a cytokinin solution. Again, that was a common mistake that I used to see quite a bit. And so making sure you divide your aliquots up, assign them to your research technicians will save you a lot of trouble. I want to talk briefly a little bit, and this covers some of the information that Chuck had, and so I won't spend a lot of time on it. But this is kind of a ranking of some of the most popular media that are used out there. And of course, Murashige and Skog is still by far the media that's used the most. DKW, which was later developed and primarily for walnuts and other woody species, is another very popular media anymore. And I think I have even heard that it's now being used quite extensively for the labs that are doing research with cannabis. Lloyd & McCowan's, again, a very low salt concentration media, but works good with many woody species. Gamborgs and Chews, more for agronomic crops, but they are also very important. Again, you have variants of your media. This is just a quick chart that kind of shows the media as just the basal stock mixture, the MS vitamins, and I want to kind of reiterate what Chuck said. One of the first things that I started working on in my career was looking at vitamin requirements for plant tissue, and that was one of the first things that I found was that the vitamins that are provided in MS media for a lot of plant species are not sufficient enough to get the best results. One of the ones that I actually started using quite a bit was the MS media with Gamborg's vitamins. You had the extra thiamine in it, and I always seem to have a lot better results using that. Now, if you're getting to the point that you want to begin to optimize your media, I've got a flow chart here that I'll go through in a moment, but what you may want to start with is some charts like this. These, and I'll quickly go through them, they kind of go along with what Chuck did in terms of presenting the different levels of ammonia, nitrates, potassium, phosphorus, and stuff like that in the media. It may be worth your while to kind of look and study these charts, figure out what you've been using up to a certain point, and then start from there as to trying to develop the best media going forward. This is, again, some of the other notable medias and what the concentrations of the ions are in those media. This was some work that was done earlier on that looked at the effects of nitrogen on the growth of the cells. And again, DKW has one of the higher concentrations of total nitrogen. WPM has one of the lowest, but you can see that the ratios are somewhat different. Okay, same thing with the micronutrients. You can look at the micronutrients if you're trying to decide on what media is best to use and start from there. You can see that most of the micronutrients in a lot of the media are pretty much the same, but there are some variations, and this is sometimes a good starting point to begin to look at. Another thing that you need to consider when you're trying to think about what type of, or how to optimize your media, is to look at the effect that pH has on the availability of nutrients. I know this is an old chart. This is back from the 50s, and it was originally developed, I believe, for soil, growing plants in soil, but it has very good applications for plant tissue culture as well. As you can see from this, certain elements are much more readily available at certain pH levels than others. And I have to be honest with you, right now we typically adjust the pH of the media to 5.7 plus or minus 0.2 or 3. Generally, I see most formulations having a pH of somewhere between 5.4 to 5.7. Looking at this chart, and when you look at the availability of a lot of these ions, I honestly think we might be better looking at adjusting the pH to 6 to 6.5. You may have greater nutrient availability in those ranges. I don't know that anyone has ever seriously looked at this, but I think it is something that you may want to, as you're trying to think about how you're going to improve your plant growth, this may be one of the things that you need to look at. Another thing, and I'm going to just very quickly talk about this, Murshigi and his work basically used ferrous sulfate and EDTA. Now, that was to help get the ferrous sulfate bound to the EDTA so it would be more available for the plants to use. Since that time, we have found there are some other sources of iron that actually make the iron a little bit more available to the plants, the ferric sodium EDTA, ferric sodium DTPA, and the ferric sodium EDDHA. All of these, while the latter two were primarily developed for hydroponics, they do work well in plant tissue culture and is something that you might want to consider if you begin to think that you're having some type of iron deficiencies. The other thing I want to point out with this slide too is that you can now find chelated forms of zinc, magnesium, and maybe a couple of other elements. To my knowledge, I don't know that anyone has done any serious work on that to look to see if these chelated forms of some of these other nutrients would be beneficial for plant growth, but it may be something that you want to consider. Again, moving on, we talked about Gamborgs. There are a number of variants of Gamborgs out there. And again, this chart basically is just kind of showing you some of the levels of NP and K between these. And again, when you're trying to make a decision where your starting point is, this may be something that you want to take into consideration. And another item that is not often looked at is the osmolality of the salt solution. Osmolality is basically going to be the ionic strength of the media. It will vary depending upon the carbohydrate source that you put in there. But again, if you're struggling to find out where or what is the best media for your tissue to grow in, osmolality may be another factor that you want to take a look at. We used to use this as a measurement to make sure that when we were producing a media that the osmolality was within a certain range for each batch of media that we made so that we knew that everything was there. In terms of stock solution and preparation of stock solutions, again, water is extremely critical. But as I said earlier, you can make stock solutions of inorganics, vitamins, amino acids, things like that. I've already kind of hit on the clean pipette between each one and putting it into aliquots, but it is important that you kind of keep some thoughts into this. Now, most stock solutions can be held for up to a year without any degradation of the material. We did a paper, it's by Hart et al., that looked at cytokinin stability. This was done probably 6 to 10 years ago now. I would recommend that you look at it because we found some very interesting things up to that point. Everybody had thought that zeatin and some of the other things were very temperature sensitive, had to be stored in the refrigerator. And through our research, we found that most of them can be stored actually at room temperature. But determining what your shelf life or your stock solution is going to be is very important so that you have consistency in your research from one phase to the next. So on this slide, part of it's missing, but on this slide, basically this is just a flow chart in terms of, you know, how you prepare your media. The one thing that I do want to point out is, again, whether you're preparing your media from a pre-made media, from you're making it from scratch, you need to add, you know, about 70% of your water at that point, add your growth regulators, carbohydrates, vitamins, things like that. Add your water and gelling agents, add your water, and then adjust your pH. Okay, one of the questions that I did have during the break was, is it really critical to adjust your pH, or how do you adjust your pH with agar in it? There are a couple of ways that you can do that. If you've got a good mixer and the solution is staying agitated very well, once you've added your agar, before you heat it up, you can adjust your pH based on that, and that is generally fairly accurate. However, if you want to be really precise, then what I would recommend is that you heat your media to right before boiling. After you've added all the components, including the gelling agents, then take your pH there. The one thing that you have to do is to make sure you clean your pH probe properly, and you also have to make sure that you're compensating for the temperature if you're taking your pH at a higher temperature. Now, most pH meters, I'm going to assume today, have an automatic temperature compensation probe that goes along with the pH, so you don't have to do anything manually. But if you're using an older pH meter, then you may have to manually compensate for the difference in temperature. Then at that point, you're going to want to dispense it into the culture vessels and autoclave them, or if you're having to pour them into Petri plates under a hood, take your big batch, dispense it into your 500 mil or however big of a container you autoclave and take under the hood to pour it into your Petri plates. So another thing that I really recommend, and this is stock solution preparation log, but this is something that we use basically in the lab anytime we were ever making anything. Whether it be a prepared media, whether it be stock solutions, whatever. This is a form that actually was passed on to me from the W. Alton Jones Science Center back in the 70s. I made some modifications to it through the years. One of the things that we did in recent years was to digitize this, and that way you can keep a digital record of your components that are going in, and if you have assigned barcodes to your raw materials as they come in, it's very easy to scan the barcode. It drops it in under the component added. You can put down your volume that you need when you actually weigh it out. If you've automated your lab and connected your balance to your computer, you can actually get the recording of what the actual weight of the product is. Same thing with recording the pH. So this is a fairly generic form. Again, it would be part of an SOP, but I do recommend that you use something like this, whether it be in paper or digital form, to record down every time a stock solution or batch of media is made. Okay, what I want to kind of do toward the end now is talk about how you would begin to optimize a media for species that you're working with. Of course, the first thing you're going to want to do is identify your species. hit the library, hit the internet, find out what's been published on this. One of the questions that we used to get a lot of times is people would call up and say, hey, I've got this, how do I culture it? You need to hit the library and see what, and again, I'm showing my age, hit the internet and see what's out there as published literature, and then begin to go from there. If you can find a formulation that has been done for this species, that's your starting point. Then you need to establish what is going to be your pass-fail criteria. Now, I don't have a pointer here, so I'm just going to have to kind of go from left to right. After you've established your pass-fail parameters, then you can test the published media. Now, this next part, and again, Max may blow me out of the water on this, and that's fine because he's younger than I am, but the way we used to optimize media is to test it 3 times. Now, you might go, why are you doing that? Well, we found over the years of research that when you're starting a new media formulation and you put the tissue on it, it may take 2 or 3 passages to either A, get components that this has been grown on in the past out of it, or to get a very accurate result. So what I would always do to my graduate students is recommend that you test the media 3 times, then average the results together. You may find that certain explants may do really well the first passage and then slowly, gradually fall off. That is more than likely the result of the tissue that you started with having a lot of the nutrients that were needed, and they have since dissipated when you put it to this new media. At that point, you can evaluate your results, and then you have a decision that you have to make. It's either failing or it's passing. It's meeting your criteria, or it's not meeting your criteria. If it meets it, great, you're happy, you can move on. If it isn't, then you've got to take a different approach. If it fails, then you can go and see if there's a different formulation that you can work with that has been published, okay? And try that and repeat the process. Or you can start to create a new media from scratch, or start to take and modify a media that is commercially available out there. Now, one of the good things about some of the media companies, and I can't say that all of them do this, but I know some do, they are now making what we call deficient media. So maybe it's just the macronutrients but no micronutrients, or it's just the micronutrients and no macronutrients. That actually gives you a base to start from to begin some of your work, and helps eliminate starting from scratch. I'm going to take the longest phase, if you will, and that's starting your evaluation from scratch. Now, in doing this, and I can't remember exactly what Miyashige used as a basal media, you're going to need to select a basal media. Because as Chuck outlined earlier, there are certain elements that are absolutely essential for plant growth. So you need to take a basal minimum media such as whites or niche and niche that's very low in salts, use that as your base media. The next step that you're going to have to do is then create stock solutions for every component that you're wanting to test. My recommendation would be to start with the macronutrients, and then after you have resolved those, then slowly work to your micronutrients. Again, you're going to need to test each macronutrient 3 times. Now, I used to use factorial experiments all the time, whether it be a single factorial, double factorial, in terms of evaluating media components. Again, Max may be able to give a little bit more information as to other ways of doing this, but however you do it, I do recommend that you repeat the results 3 times, or, take the tissue that has grown in the media for 1 generation, put it to the next generation, and then to the final generation, making sure that you use the same tissue and making sure that you go to the same media to get the best results. Again, at that point, you're going to come up with a pass-fail, okay? You need to make a decision then. Do I go back and look for another source? I think everybody knows that ammonium nitrate, potassium nitrate are some of the most common forms or sources of nitrogen, but you can also use magnesium nitrate, you can use calcium nitrate. There are other things out there that you can look at in terms of sources of some of these elements. And basically, you just keep continuing this work until you have optimized each individual component that you're looking for to get the best results that you're looking for. And again, you have to determine what those results are going to be, whether it be shoot number, shoot growth, root development, callus production, whatever. You're going to have to make those decisions, and it will vary from every lab to the next. Once you have finally gone through and determined, okay, I think these are the optimal concentrations of each individual components, then what you need to do is to make a batch of media with that formulation, but remove the base media. Okay, so if you were using a whites as your basal media, you need to pull that out then at that point. But what you have to do is if that basal media had, let's say, 10 millimolars of nitrogen in it, you need to make sure that whatever concentration of nitrogen you deem to be best in your earlier experiments, you add that back to it, or else again, you're going to be in a deficient situation. So you have to know what your basal media is, you have to know which components you're going to test and what the levels in the basal media are, and then when you make your final batches, add that back in. Then again, test that new media that you have come up with at least 3 times, evaluate results. Hopefully at that point it passes, you're happy, and you can move on. If you start to see discrepancies, things are not looking as good as what you had expected them to be, you may have to go back up and then start seeing if you can figure out how different components are interacting with each other and how that might be affecting your overall growth. Okay? Now, a little bit easier way to work from this is to start with a modified formulation. And there's really 2 ways in which you can do this. Let's say in the literature you found that the MS media was what had been used in the past, but you just can't replicate the results. So the first step I would probably do would then be to take things such as DKW, Lloyd's and McAllen's, maybe Gamborg's, try those first. See if those will work. If they don't, then it's a matter of taking and getting one of the deficient media that is commercially produced, starting with it, and then beginning to make your stock solutions for the things that are deficient in that media and begin adding them back to try to determine what is going to end up being the best formulation. This is a lot of work. I mean, this is not something you're going to be able to go and do within 30 days, okay? I know that there is some work, which I'm excited to hear about, potentially with AI development of media. I don't know how successful that will be. Max will touch on it, and then hopefully next year's meeting will get into that a little bit deeper. But you know, this is a long process. It will take sometimes a year, maybe 2 years to really optimize the media. But a lot of it goes back to what you have established as your pass-fail parameters of what you're wanting to do. So, a few closing thoughts. I hope that what I and the rest of the people on the panel have been able to do is to convey to you the attention that you need to take when developing written protocols for work in your lab and for developing new media. I hope this has given you a little bit of an insight as to how these different compounds react with each other and which is the most important when you're trying to optimize the media. Now, I'm going to say one thing, and the people in here that might be editors of the journal or reviewers of the journal may completely disagree with me. But one of my pet peeves for years has been that when you look at a research article, a lot of times they may say they use an MS media at this concentration of a plant growth regulator, and that's maybe all they say. Number one, you have no idea, did they make that MS media from stock solutions? Did they buy it from a commercial lab? Did they make their own stock solutions of the vitamins and plant growth regulators? I feel it's important, and this is not to promote any company or anything like that, but to try to get better consistency in research results, is that we at least in a footnote or an addendum put where you got your sources of materials from, okay? Whether it be your prepared media, whether it be plant growth regulators. I think that is important if you're trying to get consistency, because one of the things that I heard many times was that people would want to start to do tissue culture of a particular species, that find something in the literature, and then when they went to do it, they could never repeat the results. I think if we're more precise in what we put in our publications, it will hopefully lead to better consistency and better replication of research in the future. I want to thank you for your time and attention. Again, I'd like to thank Piero, Michelle, and the Society for inviting me. I also need to say a special thanks to Dustin Banbury and David Hart from Phytotech Labs for providing me with some of the graphics. I'd also like to thank Dave Early, the president of Advanced Ceramics, for permission to use some of his graphics in the talk. I hope you guys have a great conference and a safe trip back home, wherever that should be. And I hope that the next time the Society is in the Midwest, that you come to Kansas City. We're the home of the Chiefs, the Royals, some unbelievable museums and shopping, and the greatest barbecue in the world. So thank you very much.
Well, thank you, Ken, for this very informative talk. I think we have more or less 40 minutes before the end of this session. Max is going to go next. We might have time for just a couple of questions. And if you have additional questions, like I said, we can address them later on. Do we have any question on the floor for now? And there is the microphone there if you have a question.
Yeah, thank you very much. My name is David from Phyton Biotech from Germany. And I like the points you mentioned about the quality of the starting material. And for one batch, we are producing 70,000 liters of B5 medium from individual substances, primary vendor, secondary vendor, long tail for the QA. So from your experience, is it possible to qualify a basal salt mixture using for GMP production of pharmaceutical?
I believe you can. I believe you can set certain parameters to look at some of the physiochemical things like I talked about, like pH, the physical appearance of the media to make sure that it's properly ground or milled. That's one way of looking at it. I know it's very simple. You can also do, there's an old analysis that we used to do, a chemical analysis that would give you the actual breakdown of what is in a media. I'm drawing a blank here. It's not, huh? ICP, thank you. Inductively coupled plasma tests. That is one way of basically testing a pre-mixed media that you get from somewhere to make certain that if it says there's X millimolar of, let's say, ferrous sulfate or ferric ions in it, you would be able to determine that. That's probably the best way of qualifying it. Now, I don't know if that, again, ICP analysis is what I used to use a long time ago. I don't know if there's better ways of testing for elemental analysis in a complex mixture, but that is one way.
Thank you.
Okay, he actually brought up something about starting material, and I did have a question about explants. And how do you sterilize explants and get them in the best shape for using in tissue culture? I'm just going to throw this out to Piero and the others. This may be a good topic for next year because I know that has always been, you know, if I'm bringing something out into the lab from the field the jungle, the greenhouse, what are the best options that I can use to get that material sterilized and in the best condition. So just throw that out as a point.
That's a good suggestion.
Any other questions?
Thank you for the talk. I'm Kung from Georgia Tech. I have a questions like you mentioned that we can make some stock solution and store it. So some stock solution we can just like store in like refrigerator, but some stock we have to freeze. And then my question is like, which side or which shoot that we should search for, like which chemical or which stock we store, like.
Okay.
Yeah.
Again, I don't want to plug any specific companies, but I know at Phytotech, we did a lot of that work, and I would suggest texting or calling one of those companies that provides you materials and ask them if they have data on shelf life of stock solutions. I know that we had, and I don't want to say here because I'm afraid I'll get the dates wrong, but for most things like your integrated salts, you could store those for a couple of years. Your plant growth regulators, most of those, particularly the auxins, were good for a couple of years as a stock solution. Cytokinins, again, I go back to refer to that article that we did a few years back that really showed the stability of the cytokinins and everything. And we even did, in that study, accelerated deterioration of the components study, and we were able to show that even under adverse conditions, these would be very stable. So for the most up-to-date results, I would suggest you contact one of the companies to see if they have that information readily available. It may be available on their websites. I don't know anymore.
Yeah, sometimes it's not some, yeah, so
Right.
that's a problem. And the second question is like, it's good that you bring up the topic about sterilization, because in our lab, we also established a lot of material recently. And then have you ever think about like, you will make some kind of like initiation medium, like the medium in the beginning, which is, we use like ppm or other thing, but some material is like really contaminated with fungus.
Right.
So we think that if possible, it have some kind of like product, like fungicide or something adding already in medium, that is easier for us.
I can tell you, okay, again, this is why I think this is a good topic for next year, because you could spend 30 minutes, 45 minutes on this,
Yeah.
that one topic alone. But in my experience, of course, you've got antibiotics that you can use, but again, you have to be very careful with those because some antibiotics will literally kill the tissue in a matter of a few days. There were 3 that I remember, cefotaxime, rifampicin, and vancomycin, that seem to have the best effects of trying to control bacterial contamination without damaging the tissue. Cefotaxime particularly was probably one of the better ones, but they do nothing for fungus. Okay.
Yeah.
With regards to fungal contamination, over the years I have tried a number of different commercially available fungicides. And the one that I kind of keep thinking back to that I tried probably first was benlate, and I'm not even sure if that's on the market anymore. But with benlate, it had like a calcified clay portion to what you could get commercially available. And when you tried to autoclave it, you would get this milky white media. But because of the calcified clay, you couldn't filter sterilize it. So we never really could get very much in the way of results with fungicides. Now, again, I've been out of this now for a little over 5 years. There may have been some products that have hit the market that I'm unaware of that have greater fungicide activity but will not kill your plant. Again, that's why I'm pushing that this is probably a topic that needs to be addressed next year.
For sure. Thank you.
You're welcome.
Okay, thank you again. For the sake of time, we're going to move to Max. I will bring to the program committee the topic because I think there is definitely value. And before you start, Max, since we've been talking about mouth palpating all afternoon, there is a safe option for all of you. The red bar in the hotel has a special margarita that is called the Petri dish, just in honor of SIVB. So if you want to have a safe mouth pipetting, that's the way I will go. So now, Max, you have the rest of the time. Go ahead.
Okay, great. Okay, how do I?
Oh.
This button. Okay. Perfect. Okay. First, I wanted to thank the Society and Piro for inviting me here. I'm really honored to speak with such good company here. And I'm going to be talking a little bit about machine learning to improve plant tissue culture. I'm not only going to talk about machine learning, but that's kind of the end point of where we're going. And I just wanted to mention one thing is that earlier, Ken had introduced me as the AI guy. I am not the AI guy. I want to put that out front. I'm very much a tissue culture person. And that's why I have Mohsen Hassami's name bolded here. And if you ask him, he's also not an AI guy. He's also a tissue culture person, but he knows much more about AI. The reason I mention this is twofold. One is that don't ask me any technical questions about the machine learning or anything, because I will not know, and I'll just tell you to ask Mohsen. The other reason I wanted to mention this is that I think machine learning can be a little bit intimidating, and some of these technologies can be a little bit, they seem a little daunting. But we're getting to the point, I think, where machine learning is becoming more accessible, and I do believe in the next 5 years, you'll just ask ChatGPT what treatments to put and what algorithm to use, and it will do all the nitty-gritty for you. And so I guess I just want to highlight some of the examples. And also, I guess the other thing I wanted to mention is just that we're kind of bleeding into next year's workshop here, where we're talking a little bit more about this. So I guess this is foreshadowing for next year to some degree. But first I want to start, Chuck and Ken both mentioned this, that tissue culture is an art and a science. This comes up all the time. Any time when something's really complicated and prone to variability, people say it's an art and a science. My take on this is it's very much a science. It's just a very complex science, and that we don't have all the information yet, right? And so, a good example is, everyone says this about baking as well. You could have 3 people bake a loaf of bread using the same recipe in 3 different kitchens, and you get 3 very different loaves of bread. Maybe the altitude's different, maybe the humidity is different, maybe they're playing the different music. I don't know what's going on. But the point here is that you can follow the recipe and still get different things. And so, I would say, maybe I am an artist, but it's really a science, right? And so this is the complexity that I'm talking about. And, I guess I can't use the pointer because we have 2 screens and stuff, but really what we're talking about today is the basal media and the plant growth regulators, so the left-hand side of this. We haven't really touched too much on the right-hand side. But even on the left-hand side, you can see all the different factors. So, we talked about, and by we I mean they, but they talked about gelling agents, they talked about minerals, they talked about plant growth regulators, all those things, right? And that's one part of the puzzle. But then when we look at the other side, there's also the X plant. So, what tissue type are you using? What age was the plant? What was the mother plant coming from? To the point about the fungus, not that anyone asked me, but my personal opinion on this is the best way to reduce contamination is to have healthy stock plants. You can add all the fungicides you want, but that's really treating the symptom instead of the cause, right? If you have really healthy, robust stock plants and you bring them into tissue culture, contamination rates just drop naturally, and you don't need these things, right? And I know that's idealistic, and sometimes you can't do that, and it doesn't always work, but that's my take. But just one example of this complexity is that you can have 2 plants, you're using the same media, one person's doing it in a magenta box, one person's doing it in a stericon, whatever you want to say, and you can get different results. And this is just different things. So this is beyond the scope of this talk. We're getting into the environmental parameters and stuff, but it's really important to keep that in mind. So do you have a vented lid or do you not have a vented lid? Do you have what's the conditions you have? And this is really important because these factors are not independent. And so we've been talking a lot about minerals, but if you put yourself in the shoes of a plant or the roots of a plant, how do plants take up minerals? Any answers? Like, how does a mineral get from the basal media into the plant and up to the leaf? Pardon? Transporter? Not usually. The main way is transpiration, right? And so nutrients are taken up through bulk flow and then they go through the plant. Depends on the nutrient. Nitrogen can move around in the plant, calcium not so much, right? So it's really relying on the plant's ability to take water up, transpire, and do those things. That can't happen if the humidity is too high. And so my point with this is that you could have a calcium deficiency even though there's plenty of calcium in the media, right? Maybe you just need to reduce the humidity, increase the lights, and then it will take up the calcium, right? And so sometimes these things are interacting, and optimizing the media by adding more calcium might fix the problem. It might not fix the problem. And so we really have to keep these things in mind. And the biggest point, though, is that this really impacts how we optimize media, how we improve the system. What else do I have here? Yeah, a couple other examples of this are that nutrients also interact with each other. So, for example, potassium and calcium can compete to be taken up by a plant. And so, again, like, you can't just look at one element at a time and expect to get to the optimum situation. The other issue here is that these are not always linear relationships. So, sometimes we can have things that don't fit the pattern of traditional statistics, which we'll get into a little bit. The next thing I wanted to talk about a little bit is Liebig's law of the minimum, and this is, who here is familiar with this? Yeah, so some people. This is from the soil science world, and so I forget when Liebig came up with this, but it's essentially saying that the growth is limited by the scarcest component. And so the point here is that if we go back to what Ken was just talking about, optimizing media and going to a minimal media and then playing with one nutrient solution or one nutrient component, the problem that can arise with this is that, let's say that potassium is limiting and you don't realize this, and you start playing with nitrogen. And so you can increase the nitrogen and it won't actually improve the growth because the potassium was actually the problem in the first place, right? And so when we go through and try to optimize single factors like this, we miss these interactions and we can run into serious issues like this. Once again, like if it's another thing, then you can't optimize factors until you know what's the limiting factor, which is not always obvious. So how do we develop protocols? Ken had a good flow chart, and he's going to scold me because the example I have, we did not do it 3 times, and we really should have. He's right, I'm wrong, but that's where we are. I think we still did some improvements, so time will tell. But yeah, so there's a bunch of different ways we can do this. So the one, the simplest one. So if you're working with a new species or whatever, a really simple, and yeah, you should read the literature and see what there is. Let's say there's no literature. The first thing to do is just to test the different media, right? And so this is kind of kind of lazy because you can just buy these from Phytotech or whatever company you're working with, bring them in, make the media, grow it. It's pretty simple. In a lot of cases, you'll find out that, okay, well, this plant grows really well on MS or DKW or woody plant media, whatever, and that's good enough for our purposes, and we're going to move forward with that. That's one. The next one is to sequentially optimize factors. So again, this is an initial approach that you can take that Ken mentioned. It misses interactions, that's the issue. And like we said with Liebig's law of the minimum, sometimes you'll never actually reach the optimum, which maybe there is no real optimum in the end, right? And then for the sake of time, we'll go over each of these independently, so I'll come back to them. So like I said, many media exist. We can test them easily. This is really simple. It can be cost effective, and that's great. The main limitation that I see with this is that a lot of the media out there are actually modifications of each other, right? And so they're not that different. I mean, there is variability, don't get me wrong, but a lot of them are not that different. So from a statistical power perspective, this is not an effective way to do this, right? You're never actually going to find out if you have a really weird species that has unusual requirements. This probably won't get you to the best solution, right? And you never really optimize something with this, but it can be good. Sequential optimization, once again, this is when we're going to keep everything stable and then just change one parameter. I'd say the advantage of this is that it's simple. Statistically, it's really easy to do. You know, it's a really easy experiment to run. You just take a deficient media, like Ken mentioned, and you vary the amount of whatever was deficient. One example of this, I would say, is we were dealing, a lot of my examples would be cannabis, because that's what we work with largely, and it really doesn't like tissue culture, even though it grows crazy out in the greenhouse. But then when you put it in tissue culture, it's a delicate little plant, right? And so we started growing it on MS. It didn't like it. And so we suspected that it was a nitrogen issue because just because of the symptoms, it looked like nitrogen. And you know, when you're dealing with a plant, I think the most important thing is to really look carefully at the plant and ask what it's telling, or like really listen to what the plant's telling you. Usually the symptoms are diagnostic, and you really need to have a keen eye. And this is where the art comes in, I guess. And anyway, we suspected it was nitrogen. Fortunately, I'm not trying to plug Phytotech, but we did buy it from Phytotech, and it was a nitrogen-deficient MS media, and basically that allowed us to replace the ammonium nitrate with glutamine, in our case, and we found a marked improvement, and so we moved from there, right? The problem with this approach, though, is it misses interactions, like I said. It can lead to false conclusions, so like I said, if I start varying nitrogen, but potassium was the problem all along, then I'll never actually find that out, right? Until I go through and do potassium, but that's a huge series of experiments, right? So yeah. The next thing is it's extremely time consuming. And so if we're going to go through and independently optimize each factor, and there's 14 components, and I think there's 14 in DKW. If we wanted to optimize DKW and go through all 14, if you can imagine, especially if we're going to do it 3 times, then we're going to be there for years and years and years going through each one, right? And so, it's really time consuming and difficult. So I think, I won't spend too much time on this because I think everyone's familiar with what a factorial experiment is. But this is basically a way where you're optimizing multiple factors at the same time, right? And so in this example that I gave you here, we're just optimizing N, P, and K, which is very much oversimplified because, as we mentioned, N, for example, there's ammonium and there's nitrate, or there's glutamine, there's all sorts of things. The other factor about this is whenever you try to change anything like this, so if you've ever done a nutrient study, potassium nitrate is usually delivered as potassium and nitrate are delivered as potassium nitrate often, right? So if you try to change the potassium, how do you compensate for the nitrogen, right? So you're not actually doing an NPK study at this point. It gets really complicated. And so, no matter what you do, there's no perfect solution here. So you just have to do your best, right? But like I said, all of these nutrients come as salts, and there's another component to the salt. So you can't just vary one element without affecting other elements, right? Let me just check out. Keep this up here. The biggest problem with the factorial experiment, though, is that the number of treatments you end up doing is quite large. So in this simple experiment where we have 3 elements, so 3 by 3 by 3, so 3 concentrations of each element, all of a sudden you're at 27 treatments, right? Then you have to replicate it for statistics. And so let's say you do 3 reps, which is the very bare minimum. We're just going to keep this at like the very bare minimum that you could do. And you were at 81 experimental units. So that's a fairly large experiment just for NPK, I would say. So the pros, it can detect interactions. So if nitrogen and phosphorus are interacting with one another, you will detect that. You can develop regression models, which is nice. So you can predict the optimum, even if it's not a treatment you tested. The cons, large number of treatments, and it really limits the number of variables. So if you think about trying to do a factorial experiment with 14 components, like in DKW, you'd have thousands and thousands of treatments, which we'll come back to. The next one, and I won't spend too much time on this because I'm also not a statistician, so I'm not going to remove the mask too much. But basically, the central rotatable designs, or the DOE, you might have heard them called, it's essentially a modified factorial experiment in which they've basically more strategically set the experimental concentration. So instead of a 3 by 3 square, you end up with a sphere if you're doing a 3-dimensional thing. If you get into 4 dimensions, you can't really visualize it, but 3 dimensions is right here. And so just a quick test is basically, without getting into the math of it, this basically gives you a lot more statistical power because you can rotate the design, so you can basically use them as pseudo-replicates, and so you need fewer replicates. So instead of the cube, you end up with a sphere, and that allows you to sort of interpolate points along the sphere. And what this really does is let you use far fewer treatments and far fewer replications to do the same experiment. So again, if we're looking at this 3 by 3 test, basically What you're seeing is in the central rotatable design, or the CCD, you can see that you have 15 treatments and 20 experimental units, whereas in the factorial you had 27 treatments and 81 experimental units. And so all of a sudden this experiment that would have taken 81 different vessels is now down to 20 vessels, right? So that sounds great, right? So the benefits of this, is basically all down to the numbers and the statistical power. And so again, if you look at this, if we were to look at 6 factors, so now we're scaling up from 3 factors and now we're looking at 6 factors. You can see with a factorial experiment, you would have 729 treatments with 2,000 or so experimental units. Whereas with the CCD, you have 77 treatments and 91 experimental units. And so essentially the rotatable design now you can do with the same number of treatments. What a factorial, you can do 3 factors, you can do now 6 factors, and so on and so forth, right? So this, again, this really sounds great. You can generate cool response curves, and then you can predict the optimum combination. This is just an example from this is a fertilizer study we did in the greenhouse, so this is not tissue culture. But you can see we had a really good relationship, really good response. This is N and P, I believe, and you can see that the plant growth was very responsive and predictable. And you can basically see in the center point down on the bottom right, that would be your theoretical optimum, and then you can grow the plant in those conditions to validate your prediction, and there you go. So that's a really good way to optimize factors. However, I find that it has a lot of limitations, and I know there's some people in this society have been doing work in this and done some really good work and published some really good papers. The reason I haven't published much on this is because I find that this approach has often failed for me. And so, there's a few reasons for this. One is you really need to carefully select your center point, and so it's all based on a regression analysis, and you want to find that curve. If you don't set your center point in the right place, it kind of falls apart. It relies on relatively simple regressions. So, if you don't have linear or polynomial relationships, can kind of fall apart. If you're missing data, this is a big one for me, this is a problem. So if you're missing data from this system, all of a sudden the whole thing sort of crumbles. And so I know none of you guys have contamination in your labs, but sometimes we'll have a couple boxes contaminate, and then your whole experiment's ruined, right? And that's a big problem. And so the DOE for us, we've used it here and there, and limited success. So, I was always looking for something better, I guess. So that's where machine learning comes in. And so, again, what is machine learning? It's a big black box, is what it is. But essentially, you take your data, you put it into the black box, and it's essentially an algorithm it's basically pattern recognition, right? And so there's many different algorithms, and I don't know enough about them to get into the differences between them and everything. But basically, there's many different ways you can look for patterns within data. That will give you a prediction. You can link it to an optimization algorithm, which will predict what would be the best combination, and then you run the validation experiment, right? And so the real powerful thing about this is that, from my perspective anyway, is that machine learning doesn't really require, it's not as rigid as the rotatable design. So if you're missing some points of data, it doesn't really matter. You'll just, it'll compensate for it, and it can just do a lot of things like that. So what can it do for me, like in more real terms, I guess is the question. The first one is it dramatically reduces the number of treatments you need to test to optimize many, many factors, right? It allows quantitative and qualitative variables. This is something that hasn't come up, right? When we're talking about nitrogen level, that's a quantitative variable. So we can have, 0 to 100 micromolar and any value in between, to any, you can divide it infinitely, basically. Gelling agent is a qualitative variable, right? So you can have agar, you can have gelrite, or you can have whatever, right? And so, how do you statistically combine those two factors together into a system? And that's one of the things that machine learning can handle if you choose the right algorithm, right? Another thing that we've toyed around with is predicting optimal media from literature data, right? And so Mohsen recently, it's a strawberry project, and he looked through all of the publications on strawberry regeneration in this case, right? And basically the machine learning was able to come up with predicted media for strawberry regeneration. And what's funny about it is we're working with a company that's doing strawberry regeneration, and it was very similar to the media that they had spent the last year developing, right? And they'd been meticulously going through all these experiments, and he was able to come up to a very similar answer just from the literature values, right? And so again, this is, in 5 years from now, you'll probably just ask ChatGPT or whatever it is, and it will probably do this for you. But that's a kind of cool function. So there's all of these studies from Chuck's masters all the way up till now. You can put all of that data into an algorithm, and it will sort of spit out an optimized prediction for you that you can test. The one thing I will say is you should always test predictions. You can't just trust that, don't trust the computers, or else Skynet will come for us, right? Yeah. Oh yeah, and the last one is the sensitivity analysis, which I'll have to speed up here, or else I'll run out of time. So I'll come back to that one. So which model is best? Like I said, there's many models. It really depends on what variables are you looking at, the type of data you're dealing with, all of this kind of stuff. So don't worry about this. Work with a machine learning person. Find yourself a MOSIN. If you want to talk to MOSIN, I'll put you in touch. Yeah, don't worry about that. Moving on to a case study, so I think a good way to look at this is an example. And so this is the work of Marco Pepe. He's a PhD student in my lab, and he's been using this. The first study was to look at lighting and carbon relations in cannabis. So basically, it's using machine learning to optimize light spectrum and sugar concentration for cannabis. Oh, yeah. Yeah, so this is our lighting system, and basically it's a 4-channel LED lighting system. So we have a white diode, we have a blue diode, a red diode, and a far red diode, and then we have sucrose, right? And everything else was based on DKW media, which is the standard in our lab, and we had them in different combinations, et cetera, et cetera, et cetera. And the goal here was to optimize what is the optimum light spectrum and intensity for cannabis micropropagation and sugar concentration. So this is kind of the experiment that we set up. So we had 5 intensities of white, 9 of blue, et cetera, et cetera, et cetera. And this was all in various combinations. I forget how many treatments there are. There's probably like 100 treatments. But again, if you were to look at this in factorial experiment, this would take thousands of treatments to do, right? Even if you reduce the number of intensities of light, which you could do, it would still take hundreds of treatments to get to the same point. So don't worry about the numbers on this. Basically, this was 3 different algorithms we compared. And basically, you can see that the GRNN was the best one. And the way I'm saying it's the best is that the R squared is the best, so it has the best correlation. And don't worry about the details of that. The other thing I want to mention here, though, is that it's not perfect, right? And so you can see with shoot length, the R squared value on the GRN was about 0.98, 0.96 in the training and testing population. In the canopy surface area, it was also good. But you can see in something like node number, it was only 0.79 and 0.74, which means it only explained 75% of the variability. And so it's not a great model. It's probably still useful, but it's not as great. So we focused on the ones that worked and moved forward. The next thing, and I mentioned this earlier, is the sensitivity analysis. And this is something that I think is really useful for this, from machine learning, is that, from this, it basically takes all these factors that we were varying, and then it tells us which are the most important, right? In this case, it tells us sugar, right? So light is not as important as sugar for any of these traits. So light is, or sugar is number 1. And then when you look at shoot length, you can see number 2 is blue, which shouldn't be a huge surprise. If you're, you should be aware that blue light is very much a factor in stem elongation. Usually more blue light leads to shorter compact plants. Number 3 in shoot length is far red. Again, shouldn't be a surprise. Far red should cause plants to stretch. And then when we look at canopy surface area, it was a little different. So now red is the most important, which again makes sense from a biological perspective because red light is involved in leaf expansion and whatnot. So it all makes a lot of sense, right? But basically it tells you some interesting things. Some other interesting things here is, so for shoot length, we found, the predicted optimum was between 46 and 89 micromoles of light, kind of standard. It was really high in far red. Again, this is not a huge surprise, right? But it basically shows us that it suggests that the prediction that the machine's giving us makes sense biologically, and so it shows that the algorithm is probably working. When we look at node number, which I wasn't talking about earlier, you're seeing 160 to 200 micromoles, lower in far red, rich in red. Kind of makes sense, I guess. Canopy surface area, 140 to 219, low in far red and rich in red. So the spectrum makes sense. But the one interesting thing about this is that these light levels are really, really high for tissue culture. And if you guys are, I assume in this audience, you guys are familiar that these are really high light levels for tissue culture. Most plants that I've worked with, I don't think they would tolerate this much light, but cannabis really does it loves light in the greenhouse and it loves light in tissue culture. And this was with non-vented magenta boxes. And so it was quite amazing to us that they want that much light. And I don't think I would have thought to test that much light if I was doing this as a human, right? But the machine told us to. So we did the validation experiment. Like I said, you should never trust the machine. And so we did the validation experiment. This is our fancy light box with our multi-channel LED. It's not really that fancy. It's actually just like a shoebox thing that we modified. And basically, these are some of the plants we grew. And so when we look at the data, the prediction is the blue bar. So the top is the shoot length, the bottom is the canopy surface area. So the predicted value is the blue bar, and then the experimental value are the other bars with the error bars. So they have error bars because there were replicates and statistics. The prediction has no variability because it's a prediction, right? But basically what you can see here is that the predictions or the experimental data were not significantly different than the prediction. And basically what this shows us is that the predicted value from the machine learning was accurate. So that's a win. So the conclusion of this is, first is machine learning is awesome. I think that's my take. That's personal opinion, I guess. Another conclusion from this is that cannabis really loves light, even in vitro, which was really a surprise to us. I just did not think that in a non-vented lid that they would want like 200 micromoles of light. Yeah. Spectrum matters, but not as much as sugar. And one of the things that I found interesting on this, and maybe I can go back to this slide here. If you look at the sugar levels that they predicted, they all range from about, I think the lowest is 2 point something and the highest is 3 point something. And so, the standard in tissue culture is about 3% sucrose, right? 30 grams per liter. And basically the machine learning told us that, yeah, there's a reason for that. These decades of research were not in vain. We're using the right amount of sugar. So I guess that was just, reassuring. And so I do have another example, but I think we're out of time.
I think we're
so I guess you guys will have to wait for this one for next year or sometime. And unfortunately, this is the one where we're actually getting to the media optimization.
I think you just gave the good plug for next year for the second phase of this workshop. So first of all, thank you again, Max, for this talk. We have time for a couple of questions, if we have questions.
Thank you for a lovely talk. Unlike you, I am a machine learning person by training.
Uh-oh.
No, but I'm going to ask a slightly different, not a machine learning question, but phenotyping question. So we heard a lot about quality of inputs, how you want to make sure that everything is there, you can reproduce, that it's replicable. So when you go and start phenotyping plants, how much do you think about variability in phenotyping when your student goes and measures the size of the shoot or counts the nodes, and to what extent do you think that might actually cause problems when you're fitting your models?
Yeah, so I think it depends on the trait you're looking at. So, stem height, I think, is pretty reliable. When we get into canopy surface area, if you're using like a leaf meter, I think that's pretty reasonable. I think we were not. We were taking images, and then there's a way to like extrapolate an estimate. So then you have estimate upon estimate. So yeah, that's a bit of an issue. And you're always going to have some of these issues. So I think consistency is the best. As consistent as you can be. But I think whether or not you're 100% accurate, I think consistency. So even if our surface areas are like 10% off, as long as they're always 10% over, I think it's still okay for this application. But yeah.
Okay, we may have time for one more question if we have a question on the floor. If not, I think this concludes this first workshop